Good afternoon to everyone. First, I want to thank the organization for the invitation to this extraordinary meeting. To start, I would like to mention how my laboratory works. As you can see here, our focus is to understand the molecular mechanism involved in the pathogenesis of neurodegenerative diseases related to motor disorder as Parkinson's disease and Huntington disease. Based on this, we are developing therapeutic approach in two main fields, gene therapy and, on the other hand, pharmacological therapy, specifically studying the effect of Ravalox and small molecules using cellular and preclinical model and human samples. In the first part, I will focus on Huntington disease. The chronic expression of mutant Huntington leads to collapse of the proteostasis network due to alteration of the intracellular bandwidth involved in the protein folding, trafficking, and degradation. One of the principal consequences of this protein imbalance is alteration of the homeostasis reticulum, or ER, which causes a consequence called ER stress. On this condition, cells activate an adaptive signal transduction pathway known as unfolding protein response, or UPR. This response is initiated by the activation of a specialized stress transducer, highlighting IR1 alpha as the most conserved signaling branch. Upon activation, this sensor controls the processing of the mRNA encoding X-box binding protein 1, or XBP1, resulting in the expression of a stable and active transcription factor called XBP1S. This transcription factor regulates genes related to process that will be reestablished protein homeostasis inside to the ER. To study the functional contribution of XBP1 in Huntington disease, we generate XBP1 deletion in neurons of a mouse model of this disease. The initial prediction was that XBP1 KO will exacerbate disease progression. However, we observed that XBP1 loss in the brain lead to increased neuronal survival and decreased levels of mutant Huntingtin in the stratum of these mice. To determine the possible mechanism involved in the neuroprotective observed under XBP1 deficiency in the brain, we performed a gene expression profile analysis of the cortex and stratum of XBP1 KO mice. The result obtained revealed that the AF2 or insulin-like growth factor 2 was the major upregulated in these mice. These results were confirmed evaluating AF2 expression by quantitative PCR in the cortex and stratum of these mice. However, AF2 is the less studied member of insulin-like peptide family. Recently uncovered the importance of AF2 to brain physiology and neurodegenerative diseases. Under brain physiological condition, AF2 promotes memory enhancement and promotes hippocampal neurogenesis and also prolonged health aging. In models of neurodegenerative diseases, AF2 overexpression restored the accumulation of amyloid beta in the model of AD. In models of ALS, AF2 is differentially expressed in resistant motor neuron, and its overexpression extended the survival of mouse model of this disease. For this reason, we explore the contribution of AF2 on Huntington disease. First, we performed cell culture study to determine the impact of AF2 in protein aggregation. Cells were transfected with expression vector for the polyQ peptide fused a GFP called polyQ together AF2 or empty vector, followed by the analysis for intracellular inclusion. Quantification of the number of GFP content by fluorescent microscopy revealed a strong reduction in the number of polyQ inclusion when was co-transfected with AF2 vector. Using biochemical approach, we also determined that AF2 expression leads to a near complete reduction in the accumulation of high molecular weight of polyQ using Western blot analysis. Given these results, we decided to investigate the possible mechanism involved in the reduction of polyQ aggregation. Recently, a study has demonstrated that mutant Huntingtin can be exported to the extracellular space, suggesting a possible mechanism, the release of extracellular vesicle. For this reason, we decided to monitor the presence of polyQ in the cell culture media, and remarkable and enhanced secretion of polyQ aggregation was observed in presence of AF2 when cell culture media was analyzed using filter trap. Then we decided to explore if polyQ was inside extracellular vesicle. Using nanoparticle tracking analysis system, we determined that AF2 expression cells enhance the release of vesicles ranging between 30 and 150 nanometers, with a peak in 100 nanometers, suggesting an enhancement of exosome release. Using Western blot analysis, we found an increase levels of polyQ both in microvesicles and exosome released by AF2 expression cells. Based on the significant reduction of polyQ aggregation observed in cell of overexpression AF2, we move forward to develop a therapeutic strategy to deliver AF2 into the brain of Huntington disease mice and assess the impact of AF2 in mutant Huntingtin aggregation. We use Huntington disease transgenic mice in which we inject into stratum a virus to overexpress AF2 or empty vector. For 4 weeks later, animals were analyzed. Local expression of AF2 in adult mice result in a near 80% decrease of mutant Huntingtin aggregation, evaluated by Western blot analysis. These results were complemented with a diminution of mutant Huntingtin inclusion observed in R62 mice transgenics located treated with AF2 gene therapy. Finally, we move forward to explore the possible alteration in AF2 expression using sample of stratum from patient, Huntington disease patient from the Harvard Brain Bank. Western blot analysis revealed a significant reduction of AF2 expression in the brain of Huntington disease patient compared to the control groups. In addition, we evaluate the presence of AF2 in peripheral blood mononuclear cells from Huntington disease patients. This analysis show a robust decrease of Huntington disease, the AF2 protein levels on Huntington disease sample compared to the healthy control, which associated with a decrease on AF2 mRNA levels. Since AF2 is a soluble secreted factor, we decide to measure the quantity of AF2 in plasma from Huntington disease patient using ELISA. This analysis reveals a significant decrease in the amount of AF2 in plasma sample from Huntington disease patient compared to the control subject. At physiological levels, previous studies have shown that this factor decreased during aging in a hippocampus region and has been proposed as a factor that could be involved in rejuvenation process of the brain based on its role in process as synaptogenesis and neurogenesis. However, the possible protective function of AF2 during aging has not yet been explored. For this reason, we decide to investigate the significance of AF2 signaling in the progression of aging and its relation to motor decline and synaptic function in neurostriatal circuits. To evaluate the effect of AF2 in neurostriatal circuit involved in motor control, we inject young animals or old animals with adeno-associated virus to overexpress AF2 into substantia nigra. After 6 weeks, we determine motor performance, dopaminergic innervation, and dopaminergic neuron, and also synaptic marker. First, we determine motor coordination capability using Rotarod test. All mice show a decrease of motor coordination capacity compared to the young mice, but this difference was not observed in the group injected with AF2 gene therapy, here in the blue bars. This result showed that the AF2 overexpression into dopaminergic neurons improved the motor capability in the old mice compared to the control groups. After this, the state of dopaminergic neuron was determined by Western blot analysis of tyrosine hydroxylase levels, which is an enzyme involved in the synthesis of dopamine in substantia nigra. We observe similar tyrosine hydroxylase levels in both groups, suggesting that AF2 gene therapy doesn't modify the abundance of dopaminergic neurons. However, this treatment could affect dopaminergic innervation to the striatum region. For this reason, we evaluate the tyrosine hydroxylase expression levels in this region and the DARPP32 enzyme present in striatal neuron using Western blot. The result again showed that the AF2 didn't modify the protein levels of both TH or DARPP32. Finally, we explore whether AF2 gene therapy could modify synaptic protein such as SNAP25 and BAM2 in both regions. Our results show that the expression of AF2 didn't modify the protein levels of presynaptic protein SNAP25 or BAM2 in substantia nigra. However, we observe a clear increase in the expression level of SNAP25 protein in the group of animals treated with AF2 gene therapy in a striatum region. According to this, AF2 overexpression could modify synaptic connectivity in the striatum region, improving the capacity of motor coordination in aged mice. So, our result suggests that AF2 expression prevents the motor impairment during aging. AF2 exerts a neuroprotective effect during neurostriatal degeneration during aging process. Treatment with AF2 increased SNAP25 levels into striatum region of aged animals, and the possible mechanism involved to explain the anti-aging effect of AF2 on mutant motor control is still unsolved. Finally, I want to thank the people who work with me studying, assistant and postdoc, and also the funding which support this research. Thank you.